The audio you're about to hear was recorded in Anchor. Learn more at anchor.fm.
From ScienceSortOf.com, you're listening to Science Sort of.
Hello and welcome to Science Sort of. This is episode 332, and here to talk to you about things that are science, things that are sort of science, and things that wish they were science is just me, Ryan Haupt. Sorry we've been off the air for a little bit. I know a lot of podcasts take a break around the holidays. We are sort of doing that, but we also sort of got interrupted by me defending my dissertation. So I am now done with grad school and with being a PhD candidate, and now I'm just Ryan Haupt, PhD. So that's pretty fun. I've been really excited about that, but I also got a new job that I'll talk about later on, and just sort of had to hit the ground running and been busy with the holidays, and it's been a tough time for everybody. So for all the folks out there listening, hope you're doing well. Sorry for the delay in episodes. We have an Anchors Away episode for you today, an anthology, but we have more content recorded that's going to be coming at you in the new year. Super excited. Hopeful about 2021. Hopeful about the vaccine getting out there. Hope everybody's doing okay. Should be a pretty fun Anchors Away. I'm going to talk about some Up-Goer 5 stuff that I was doing with AGU at their annual meeting, which just happened. So it's kind of a good refresher for that. Talking about whales, talking about dinosaurs, talking about dogs. It's going to be a lot of fun. So I will jump back in a little bit to tell you about what I'm drinking, and then we'll round out the end of the episode with a quick PaleoPOW and let you on your way to enjoy the holidays, enjoy some rest, and enjoy the upcoming new year. See you in a second. Today I thought I'd try a little experiment here on Organized Curiosity, the anchor station devoted to cataloging everything we know about the natural world for your edutainment. I'm your host, Ryan Haupt, and here's the experiment I had in mind. When scientists are working on research, a lot of times we'll have preliminary data or something that's in progress that we're working on making into a paper, and so we'll submit it as an abstract or short summary to a organization that's having a meeting. So for example, as a paleontologist, a vertebrate paleontologist in particular, I try to go to the Society of Vertebrate Paleontology's annual meeting every year, which means that I have to submit an abstract to that meeting to get the opportunity to present my work as either a talk or a poster. I'm actually in the process of working on my abstract for that exact meeting, but I'm taking a break to record this. Last year, one of the meetings that I like to go to, the American Geophysical Union annual meeting, had a cool session. So the way it works sometimes with these meetings is you can submit an abstract to a specific session that's centered around a specific theme. And they had a session that was dedicated to writing your abstract using what's called Up-Goer Five speak. This is based off of an XKCD comic that Randall Munroe did, where he described a Saturn V rocket using only the 1,000 most commonly used words in the English language. It's kind of a cool exercise, and it emphasized some of the struggles that can be had when trying to describe complex or technical things using only common words. The exercise was so successful that lots of meetings have actually adopted the same sort of format for a session, and Randall was actually able to put out a book called Thing Explainer that you can get online, which has more than just the Saturn V being described using the same language. So I thought it would be fun to try to submit some of my own science, or at least the idea of my science, in this format. I've taught writing courses in the past to graduate students, and this is actually an exercise that I put them through to ask them to simplify the way that they write about their science. And I don't think it's a perfect way to simplify the way we talk about science, but I think it is illustrative in showing some of the pitfalls, or at least some of the blind spots we might have when it comes to the jargon and technical language that we rely on to communicate with each other that might make science less approachable to people outside the sphere. So with that, I'm going to come back in just a second and read my abstract that I submitted to the American Geophysical Union meeting, and then I'm going to try to translate that into actual science talk, and we'll see which ends up being the better example of science communication as voted on by you, the listeners to this station. So stay tuned. I'll be back in just a moment with some very convoluted science. All right, stay curious out there. There's a lot to learn. right, it's Ryan here from Organized Curiosity, and just a reminder of what we're doing today. I'm reading an abstract that I submitted to the American Geophysical Union's annual meeting last year, where I had to describe one of the science research topics that I am interested in using only the 1,000 most common words in the English language. And now I'm going to read that abstract to you to see if you can make sense of it. The title was Using Very, Very Tiny Things That Are a Bit Heavier Than Almost the Exact Same Very, Very Tiny Things to Understand Much Bigger Things Like Animals. Here's the text of the abstract. Everything that is made up of very, very tiny things. You are made up of these things, and so is the air you breathe, the water you drink, and the food that you eat. There is an old saying, you are what you eat. This saying is true. Sometimes the very, very tiny things that make us all up are a little bit heavier than normal. Sometimes being more heavy is bad, and if you hang out with it for too long, you will get sick and die. We are not talking about that type. Our type is totally safe, just a bit heavy is all. Being heavy does not change what the thing is, but it does change how the thing moves around the world. And if we look close at things like animals, we can learn about what went into making them that way. We do this using state-of-the-art boxes with lights and computers and other stuff inside. We can figure out how much of a thing is made up of the very, very tiny things that are a bit heavier but still pretty much the same as the other very, very tiny things. Why does this matter? Because sometimes we want to know if an animal ate other animals or if it ate things that grow out of the ground that are not animals. Why not just watch what the animal does? Because sometimes the animals are hard to see because they live up in the trees or deep in the water. Other times the animal has been dead for a long time, so long that it might not even have any family left. It's sad, but it happens. Turns out, the numbers the box give us are from the body of the animal that was doing the eating, so we can know what it was eating that way, and we don't even have to watch it. It is important to know a lot about the box we are using, and also about the animal and types of food the animal might have eaten, but it's still a great box for learning about animals and all sorts of other things too. I work a lot with an animal named after one of the 7 bad things we're told not to do, the one where you don't really do anything at all, you just sit there doing nothing. Maybe eating sometimes, but that's a different bad thing than the thing I'm talking about today. Today they live in trees where it rains a lot, but there used to be Today they live in trees where it rains a lot, but there used to be different types of this animal that lived on the ground and in the water once. What were they doing all day? Good question. But to answer it, we need to learn about the ones that are in the trees today too. Even though they are different, they are still family. I hope what I learn helps keep the animals around and up in the trees for a good long time. All right, so if you think you have an idea of what it is I was actually talking about there, please call into this station and let me know. I'll be really curious to see how this actually played with a general audience, because so far I've only read this to bemuse scientists who chuckle at the linguistic hoops I have to jump through to make this work. So make sure you favorite this station so you will be alerted when my response to what this actually means goes live here shortly. And until next time, stay curious, because there's a lot to learn out there. Ryan Haupt here for Organized Curiosity, and today I've been doing a bit of an experiment where I read an abstract that I wrote using only the 1,000 most common words in the English language, and in my opinion it's a bit convoluted and doesn't necessarily get the point across that I was trying to make unless you already knew what the point was or had visual aids. So this abstract was accepted to the meeting, and I had to give a 5-minute presentation where, again, I was only allowed to speak using the 1,000 most common words, but I was allowed to have a PowerPoint presentation up on the screen. So that obviously helps because you can show the thing you're talking about, even if what you're saying is in very convoluted terms. But what I've yet to attempt is to reverse engineer my own abstract back into actual scientific jargon and lingo to see if I can even make sense of it and if it has the same kind of flow I intended it to have when I first wrote it. So without scripting this out ahead of time, I'm just looking at the text of the abstract I just read in the previous segment. I'm going to try and re-say it with proper science talk. And if you were getting ready to call in to see if you could guess what the abstract was about based on the previous segment, this is a spoiler warning because I'm about to reveal what it was actually about. Everything is made up of atoms. You are made up of atoms, the air you breathe is made up of atoms, the water you drink, and the food that you eat. They're all atoms. There is an old saying, you are what you eat. This saying is true. Sometimes atoms are a little bit heavier than normal. We call these isotopes. Sometimes being more heavy is bad, so those are radioactive isotopes, and if you hang out with them for too long, because they're radioactive, you'll get sick and maybe die. I'm not talking about radioactive isotopes. I'm talking about stable isotopes. So they're totally safe. They don't radioactively decay. They don't emit any radiation. They're just a bit heavier than the normal form of the atom. That's what I mean when I say that being heavy doesn't change what it is, because it still has the same number of protons and electrons. So it's still the same atom. So it's still a carbon or an oxygen. It's just instead of being an oxygen that weighs 16, it's an oxygen that weighs 18. So because chemical reactions rely on energy, heavy atoms move through chemical reactions differently than their lighter counterparts. So even though they're chemically the same, the physicality of how they move around the world is different. This difference can be reflected in the tissues of animals dependent on where they live, what they drink, and what they eat. So we can actually measure the tissues of animals, and the ratio of the heavy to light isotopes in those tissues can tell us something about their diet, their water source, their geographic location or movement across the habitat, all sorts of things like that. We can measure the stable isotope ratios using what I called state-of-the-art boxes with lights and computers, but what I would actually call a stable isotope mass spectrometer. Why does making these measurements matter? So in the case of something like nitrogen isotopes, we can use those nitrogen isotope ratios as an indicator of where the animal fed within the food web. So if it was a carnivore eating other animals, or if it's an herbivore eating plants, or if it was an omnivore somewhere in between. The nitrogen isotopes can tell us that. Why is this better than just observational studies? Because sometimes animals are really, really tough to see or track. So for things like whales that live out at sea and are really difficult to observe all the time, or things like sloths, which live up in trees and don't actually do much that often, it can be easier to just take tissues from those animals and analyze those instead of putting in all the time and energy just trying to observe them directly. This is also really useful in the case of extinct animals, because extinct animals can't be directly observed, so you have to use these other sorts of proxies to figure out what they were doing, especially in the case where they don't have any living analogs. So I look at ground sloths, there's nothing really like a ground sloth in the environment today, so we need to use other techniques to figure out what they were doing. And that's the joke I was making with the animal named after one of the things we're not supposed to do. I was saying the 7 deadly sins, one of which is sloth. They do also eat sometimes, but they're not gluttons. They live in the rainforest, but there used to be all different kinds of sloths that ran around on the ground, and even a marine sloth that lived in the water. We're trying to figure out what they were doing all day, but a part of that question can be answered by looking at the stable isotope values of their living relatives, the tree sloths, and then using that as kind of baseline data to go back in the past and figure out the ground sloths. Ultimately, our hope is to use these techniques to better understand how these animals respond to climate changes and perturbations, because at the end of the day, all of these sloths are still one big group of animals, and the hope is that by better understanding them as a group, we can help make concerted conservation efforts to keep sloths around for a long time to come. So that's the abstract I would have written if I was just writing the way that I tend to speak, but I was limited by the 1,000 most common words, and now you can kind of see the difference, and hopefully I would guess this version made more sense to people, but maybe it didn't, and I'll be fascinated to hear some feedback from y'all, so call in if you have a hot take on what you just heard. Thanks for listening. Make sure to favorite this station for more content like this every day, and until next time, stay curious. There's a lot to learn out there. So to wrap up Up-Goer 5 Day here on Organized Curiosity, I, your host, Ryan Halp, am going to do a little bit of reflection on this exercise and what I think it kind of means for science communication and how scientists talk to other scientists versus how scientists talk to the public. I think forcing scientists to write in the Up-Goer 5 language is a useful exercise to get scientists thinking about the way that they communicate. I don't necessarily think that it's the best way to actually communicate. I think the silliness of it can be amusing, but not necessarily informative. So what I mean by that is it's amusing to watch scientists kind of dance between the raindrops and avoid relying on the words that we become so comfortable using as part of our jobs. But I don't know that it's actually informative. So saying that you study, temperature changes in the big, big blue water is amusing, but isn't it more clear just to say ocean? Also, the word sea, S-E-A, not the letter, is in the 1,000 most commonly used words, which got me thinking about how in some of the segments I do on this show, like Better Know a Root, rely on a good understanding of Latin and Greek, but English is a Germanic language, meaning a lot of the most common words that we use in English don't have Latin and Greek roots. They actually come from older Germanic sources. And so a lot of our 1,000 most common words are coming from that. And so there's this bias against science words because science words tend to be the more highfalutin version of a given word. So sea comes from a German root, whereas ocean comes from a Greek root, I think. So there's a little bit of a bias there in that just the way science is typically spoken about and written can't be used because of the limitations of the exercise. So that's one con. Another is I would argue that jargon in science can serve a purpose. There's a reason we use the words that we use, and it's because typically the words that we use, even if they seem jargony in the same way a physician would use them, it's the most succinct or precise way to explain the concept that we're saying. Yes, it does mean that you have to know what that word means in order to understand what's being said, but amongst scientists, we usually do. The problem is when we speak that way to the public without thinking about how difficult those jargony terms might be if you weren't already familiar with them. The things I like about the Up-Goer 5 challenge is that it provides really clear limits. It makes you use really unambiguous words for the most part. One example I've seen where a word that is a common word but maybe can be used in a science context is fault. So we would say someone's at fault for causing a problem or doing a bad thing, but a geologist would use fault in the term of a plate tectonic boundary. And so you can get away with saying fault in the Up-Goer 5 language, but your meaning might be more ambiguous than you intend. I think that's a rare case, but it is something I've thought about in approaching this exercise. Another cool thing about this exercise is that we tap into pop culture a little bit. So sometimes scientists are a little bit aloof. They don't necessarily want to play the pop culture game. But, this is taking a joke that a webcomic made and kind of riffing on it and having a good time with it. And I think that that's not a bad thing for scientists to think about trying from time to time when trying to connect with the public. And then the ultimate conclusion I kind of came to is that I see it as both a pro and a con, but this exercise forces researchers to think differently about the way they present their work. And I think that that's almost always a good thing. It takes time and energy that could be spent doing other things, but sometimes that self-reflection is needed and isn't always done. And in a field that is constantly having to justify its existence to the general public and government officials who might not want to give money to science, I think it's really important for scientists to think about the way we communicate and make sure we're doing a good job to get our message across so that everyone can get excited about science. So if you enjoyed me kind of going through this exercise today, I would really appreciate any applause or echoes you feel like handing out, and be sure to favorite this station for more content like this every single day. Until next time, stay curious out there. There's a lot to learn. Hello and welcome to Organized Curiosity. I'm your host, Ryan Haupt, and today it's news you probably cannot use, except this time you might actually be able to use this, so pay attention. Cheerios is sending out packets of wildflower seeds in an attempt to help make sure that bee pollinators have wildflowers in their area to keep those populations healthy. So this seems like it should be a no-brainer, right? Plant some flowers, help some bees. But there is, unfortunately, a problem, and I hate to be that guy who's just like, well, actually, this is not as helpful as you think it is. But actually, it's not as helpful as you think it is. Because Cheerios are being a little bit indiscriminate in the way that they're trying to get the average consumer to help out with this problem. And it is indeed a problem. Bee populations are declining thanks to this thing called colony collapse disorder, in addition to a general loss of habitat and use of insecticides and herbicides that both hurt the bees and hurt the plants that they would use to feed on. So the seed packet that you might get sent from Cheerios actually contains a number of plants that could be considered invasive in your local area, to the point of being considered banned. For example, the forget-me-not, which I believe is Alaska's state flower, is not legal to plant in Massachusetts and Connecticut. You can't just send a packet of nice seeds that is going to apply equally well to every area of the United States. Catherine Turner, who's an ecologist who specializes in these kinds of things, said first and foremost that when it comes to planting a flower, context is important. Here's the quote. No plant is inherently bad, but many species can and have caused a great deal of damage when they are introduced into locations outside of their native range. Invasive species can outcompete the natives they encounter. They can take up all the space and use up all the resources. It doesn't happen with every plant and in every location, and scientists like me are working now to figure out why that is, how to predict what will cause a problem, and how to manage or prevent invasions. So I'm not trying to say that this is altogether a bad thing. It's just you have to be very careful when planting seeds that you're not 100% sure about in your backyard. There are ways to find out if the seeds that you have are actually fine to plant. There's a website called Xerces, xerces.org, which will tell you all the native plants to your region, including publishing regional gardening guides to help you figure out the best plants for a DIY approach. The U.S. Department of Agriculture also maintains a USDA plants database where you can look up a specific plant and find out if it's native to your state. You can also look at the legal status to see if the plant is on any federal or state noxious weed lists. A big part of this push is being sold as a way to help honeybees. And honeybees are obviously an important insect in our food economy, but they're just that. They're actually an agricultural kind of product and are pretty carefully maintained by the people whose job it is to distribute honeybees around the country for various pollinating seasons. This is an actual business that exists. Bees get trucked around the country and let out in various areas to help pollinate during that specific pollination season. I remember this being a big thing in California when I lived there. And so those bees, while they're still at risk from things like habitat loss, because they're more of a domesticated, managed thing, it's not as bad. The worst situation is for native bees. And a lot of those native bees are solitary, subterranean, and don't even have stingers because they don't have a colony or a hive to defend. So one of the things that's actually really important is to make sure that in areas that are really dense with different agriculture, that you also have areas set aside as bee habitats where you don't spray herbicides, you don't spray insecticides, and you give those local bees space to kind of pollinate those plants and do their thing, and as well as space to dig their tunnels and lay their eggs and continue their life cycle. Now, fortunately, General Mills is actually helping with that. They are making sure that the areas that grow oats for Cheerios, which oats are wind pollinated, so they don't even need the bees, and they're making sure that they plant dedicated pollinator habitat near or right next to where they're growing the oats for the Cheerios. So that is a good thing. I am in no way trying to impugn General Mills' efforts and intentions. It's just a little bit more complicated than maybe it seemed at first. One last thing I'll plug, because I saw it online earlier today, is there is a web company called Seedles, Seedles, that will send you a seed ball, kind of like those bath bombs, but full of seeds, and those seeds will be specifically tailored to your region. So that's another way to go get some seeds if planting plants in your area that will make the bees happy is important to you. Let me know if for once this news was actually useful to you by calling into this station, and remember to stay curious out there because there's a lot to learn. Well, it would not be an episode of Science Sort of if we didn't talk about what we are drinking. And I am drinking a festive holiday beverage. It is called glögg. It is a Swedish mulled wine. And my wife Julie made it, and it's delicious. And it's red wine with mulling spices, and you eat it or drink it with some brandy-soaked raisins and almonds, so you get a little protein, a little fruit. And the cool thing, this is so dumb, the raisins. Julie asked me to buy some fruit to just have for snacks around the house. And so I saw grapes, they were on sale. I was like, oh, I'll get her some grapes. Grapes are great. Turns out she's not the biggest fan of grapes. So the grapes just kind of sat there in the fridge for a while. Grapes, turns out, last a pretty long time in the fridge. But then they're starting to go, I could tell. And I was like, oh man, what do we do with all these grapes? And I was like, what if I just make raisins? And I put them in the oven at 200 for hours. And eventually we have raisins. And then we soaked them in brandy so that we could have glögg. And they plump back up to like half their original size, but just full of brandy instead of water, I guess. And they're delicious. Homemade raisins. Who knew? Absolutely fantastic. I think the recipe for the glögg is on
the website that my mother-in-law maintains for all of her recipes. So if I can find that, yep, I found it. I will put it on the website for this episode, and you can enjoy that. All right, back to the content.
Very much. I'm listening to you in Cork in Ireland late on Sunday evening, and it's my first day on Anchor version 2, and I must say I'm very pleased to find your station, because popularizing science, talking about it in a way that makes it understandable to somebody who doesn't have a scientific background is music to my ears. I've got a daughter who's 11, who I hope one day will take science very seriously. And I'm also a great fan of whales. Not that I've ever met one. Thank you.
Time for a quick Q&A, this time sent in from Paul from Cork, Ireland. Paul wanted to thank me for doing the station, which I always appreciate, and mentioned that he hoped his daughter would become interested in science. And that reminded me of a point I like to make with people, and it's that as I do science outreach, if there are young people listening who don't know what they're going to do for a career yet, yes, science should maybe be on your radar. I think it's a great job. The best thing I think you can do is try to get work in a lab as an undergrad, which is what I did, and helped me realize whether or not this lifestyle, as well as the aptitude for it, was something I had and felt like pursuing. But I also don't want to try to convert people to go into science just because it's what I do. It's a specific type of career and profession, and it's not necessarily suited for everyone. But sort of like stand-up comedy or being a chef, you don't have to do it professionally to still appreciate it. So you can still laugh at a comedian's joke and eat a chef's food, and I think the same should be true for science. I don't want everyone to be a scientist, but I do want to live in a society where people are generally scientifically literate and appreciate the value of having science done as part of their society. So hopefully that's like a reasonable middle ground in terms of evangelizing the science that I do. I also wanted to touch on what Paul mentioned about not knowing any whales, because that reminded me of a story. We were talking about humpback whales and the supergroups they're forming, and I didn't talk about it in the segment, but I actually have been up close and personal with a humpback whale, which was one of the more amazing experiences in my life. So I did want to kind of touch on it and also touch on a little bit more about humpback whales. I was helping out with a summer science camp when I was an undergrad at the University of California, Santa Cruz, which is on Monterey Bay, home to many, many different species of marine mammals. And one of the things we got to do as part of this summer camp was take the kids out on a whale watching cruise. And while we were out on the whale watching cruise, we saw so many different kinds of marine mammals. It was incredible. I mean, we saw huge pods of dolphins. We saw whale spouts, off towards the horizon. And sometimes you can even tell which type of whale you're seeing based on the shape of the spout from the blowhole. But the coolest part for sure was when a juvenile humpback whale, so, like only 40 feet long, got really curious about our boat and swam right up to it. And because there are laws like the Marine Mammal Protection Act, and because we were on a whale watching cruise that was conscientious towards the health and safety of the whales, as soon as the whale approached within a certain distance, the captain killed the engine and basically wouldn't turn the engine back on until the whale lost interest and swam away. And these are curious animals and they're stimulated by their environment. So we got to spend probably about 40 to 50 minutes with this one whale just swimming underneath our boat back and forth, going from side to side. One thing you learn about humpback whales is that because they have these really large pectoral flippers, they are actually extremely maneuverable in the water. So this whale never bumped our boat once, even though it was only a couple of feet away. And it would swim under and it would kind of rotate to the side and its one eye would kind of look right up at us. And then it would spout from its blowhole and then do a 180 and go back onto the boat and kind of do the same thing. And it just kept doing that over and over again. And while it was incredible, I also learned that whale breath smells terrible. We were getting moisture from the blowhole sprayed on us as the whale was doing this, and it was so foul and terrible smelling, in part because this type of whale, humpback whale, is a mysticete, which means that it has baleen instead of teeth. So it kind of has this hairy comb in its mouth. And the way they feed is they will lunge into a group of fish or squid or large zooplankton, and then they'll take this huge mouthful of water that also contains all the food that they want to eat. And then they have this really powerful tongue, and they'll use their tongue to push the water back out through the baleen, filtering out the food. And the food gets trapped in the baleen as the water gets pushed through it, and then they swallow all the food, and that's how they feed. But because they don't have hands, they never brush the baleen. So everything that gets trapped in there and doesn't get swallowed just basically rots inside their mouth. And it was just so bad smelling. And I've had experience with other marine mammal bones, and marine mammal bones also smell bad. They eat so much fish and squid and other types of seafood like that the oils from those animals like infuse the bones. And so the bones themselves are kind of this greasy, oily, fishy smelling thing. I love marine mammals. I think they're amazing animals, but that part of it was slightly less pleasant. So thanks again for calling in, Paul, and giving me a chance to talk more about whales. I always appreciate that. I always appreciate you guys listening, applauding, echoing, calling in with whatever you have to tell me. And with that, I'm going to sign off for the day. But remember to stay curious, because there's a lot to learn out there. Hello and welcome to Organized Curiosity, the Anchor Science Station dedicated to cataloging everything we know about the natural world for your edutainment. And it's kind of a special day because we might have to recatalog something we thought we had understood for a long time. And that's how dinosaurs were related to each other. So before I talk about what this new research is proposing, I thought it would be good to go back and take a brief look at what we thought about dinosaur relationships prior to today. So in 1887, the group Dinosauria was divided in twain by British paleontologist Harry Seeley based on one striking difference between the two traditional groups of dinosaurs, and that was the structure of their hips. So the two groups are named after those structures. One group are the Ornithischians, and the other group are the Saurischians. Now, Ornithischia literally means bird-hipped, and Saurischia literally means lizard-hipped. This is going to get a little confusing, so I'm going to try to be really clear with the terminology. So in bird-hipped dinosaurs, we have the pubis and the ischium in alignment with each other, whereas in the Saurischian pelvis, the pubis is facing forward and the ischium is facing backwards. And it's a pretty clear difference when you look at the hips side by side, so it seemed a pretty good way to delineate between these two major groups of dinosaurs. The Saurischian dinosaurs include things like the sauropods, which are the long-necked dinosaurs, Brachiosaur, Apatosaur, Diplodocus, and also theropods, which are the meat-eating dinosaurs, things like Deinonychus, Velociraptor, and T-Rex. Now, where it gets even more confusing is the fact that birds evolved from theropods. So birds evolved from the lizard-hipped dinosaurs, not from the bird-hipped dinosaurs. Bird-hipped dinosaurs are completely extinct. So the terminology there has always been a little bit confusing, but it's been pretty well established that this is the way that dinosaurs are related to each other up until literally today. So I think that's a clear way of describing that distinction in an audio format when you can't actually visualize it. And hopefully that gives everyone the background that we need to move forward to talking about the research that was published today. Coming up in just a minute here on Organized Curiosity. A paper published today in Nature, which is one of the most prestigious journals in the world, authored by Matthew Barron, David B. Norman, and Paul M. Barrett, titled A New Hypothesis of Dinosaur Relationships and Early Dinosaur Evolution, has the world of paleontology talking, and probably continue to talk for quite some time. As I mentioned in the previous segment, dinosaurs have classically been divided into two groups based on the structure of their hips. Now, most of the dinosaurs you're probably familiar with are actually late additions to the dinosaur family tree, and in part that just has to do with how evolutionary radiation works. When dinosaurs first began, they weren't as diverse as they would later become, but it also has to do with this process of fossilization, preservation, and later discovery being biased towards more recent things. So the more recently something died and was fossilized, the more likely we are to find it, and the more likely we are to find it in good shape, intact, with most of its details still preserved. So our early knowledge of the earliest dinosaurs is actually pretty limited. It's thought that dinosaurs first evolved around 231 to 243 million years ago, which is during the late Triassic. And it was also thought, based on where our earliest fossils are found, that they evolved somewhere in the southern part of Gondwana, one of the two halves of the single supercontinent that existed on Earth at that time. At the end of the Triassic, there was a major extinction event, one of the big five mass extinctions that have happened in Earth history that we're aware of. The earliest dinosaurs survived this extinction and went on to then diversify into all the newly available habitats that had been previously inhabited by earlier versions of other kinds of reptiles, but were now empty because they'd gone extinct. So dinosaurs have these opportunities to go crazy. Adaptive radiation is the term we use for it in evolution. The paper that just came out in Nature is suggesting a pretty substantial rewrite in the earliest part of that story. So the way this kind of research works in general is if a person wants to reconsider the evolutionary relationships between fossil animals, is they take a bunch of fossils that are representative of the taxa that they're interested in and a few groups that are outgroups of that group. So you get kind of a inside and outside perspective of what's happening, and then you pick a bunch of traits that are evolutionarily divergent over time. You measure those traits very, very carefully, and then you use sophisticated computer algorithms to basically rebuild the evolution that would have had to have happened to get a group of organisms with those traits in those configurations as you measured them. And what the algorithm does is it tries to find a solution to that puzzle using the fewest steps possible, and that's called parsimony. So basically, it doesn't make sense to argue that something like a sea lion and a seal evolved completely separately. It's possible, but it's more parsimonious to say that there was a single lineage of bear-like animals that entered the water and diverged into things like sea lions and things like seals. What the first author of this paper, Barron, did that hadn't been done before is he built a data set based on 74 species of dinosaurs and dinosaur-adjacent species towards the base of the dinosaur family tree. So he tried not to focus on things that came late in the game and really just focused on those earliest representatives that we have that show us the beginnings of dinosaurs. And based on his analysis, he actually pushes back the date of dinosaur first arriving on the scene to 247 million years, so about 4 million years earlier than the earliest previous estimates, and suggests, based on the biogeography of where the specimens that he analyzed were found, that they probably actually evolved in the northern half of the supercontinent Laurasia. So those are two really cool results, but the biggest result that he got was that no matter how he put the tree together, the way that it made the most sense was to put the theropods, the meat-eating dinosaurs, with the ornithischians, not with the sauropods. So he's actually saying that this hip structure difference isn't the defining feature that we should be using to separate out these two major groups of dinosaurs. So now we have a group called saurischians, which only contains the sauropodomorphs, the long-necked dinosaurs, and a new group called ornithosclidians, which contains theropods and all the previous ornithischians. This is a pretty substantial rearrangement of all the dinosaurs that we know, it doesn't change what we know necessarily about their lifestyle and ecology and behavior and functional morphology, but it does change how we thought that they were all related to each other, and it does change where things like the origin of feathers, which is really important for the story of how birds evolved, because there are ornithischian dinosaurs that actually had feathery-like ornamentation that we found in the fossil record, and putting them together with theropods actually makes a little bit more sense from that perspective. One point that Barron was really careful to make was that there is no one single trait that you can use to separate out the two groups, as had been done previously with the hips. It's now a much messier story with 21 different, harder to talk about features that characterize the new groups that he is saying dinosaurs fall into. This isn't cut and dry. This isn't totally the end of the story. It's obviously a really big deal. It's something that's going to require a lot of discussion. It's going to require a lot of people to go back, double check his work, rerun the results, come up with their own tests, and if all that passes muster, it's possible we might have just lived through what's called a paradigm shift, which is when an entire branch of science has this watershed moment where everything kind of changes, and it stays that way as the field moves forward. And it's a big moment. And it's really cool to see it happen in real time. It's really cool to see my friends online who aren't paleontologists or even scientists get excited about it. So if you're excited about it, if you have a strong opinion on whether or not you buy this new arrangement of all your old favorite dinos, let me know by calling into the station. And remember to stay curious out there, because there is a lot to learn still. Obviously, we just learned something new. It's going to happen again, probably. Basically, what I told one of my friends today is don't come to science if you're looking for facts that never change. That's never going to work out well for you. All right, bye. Hello and welcome to Organized Curiosity. I'm your host, Ryan Haupt, and today, since it's International Puppies Day, I figured we could start with a bio for the pupper we're celebrating today, Canis lupus familiaris, the domestic dog. Obviously, there's no way to fit everything we know about man's best friend into a single segment, so I'm just going to hit some highlights and hope that you are curious for more down the line and ask about it with Collins. So, let's talk about the word dog, because it's kind of interesting in that we don't actually know where the word dog comes from. It is derived from Middle and Old English, dokga, which was thought that was maybe a Proto-Germanic term for something that was really powerful, strong, and muscly, and with a diminutive suffix ga, making it littler, so like a little powerhouse, a tiny ball of muscle. But that's just a theory. We don't actually know. The word just kind of shows up in English and is a legitimate mystery how it first cropped up, because if you think about languages that are closely related to English, like German, the word is Hund, and that's where we get our word for hound. So it doesn't come directly from our single common ancestor with the German language. It's a word unique to English that just appeared, which is pretty cool. The appearance of dogs as a domesticated animal are also not quite clear. When I gave the scientific name earlier, I used 3 words instead of my normal 2. So I said Canis lupus familiaris, and that's because dogs are a subspecies of gray wolves, Canis lupus. But gray wolves have a very wide distribution all around the world where people live, and so it's hard to pinpoint exactly where dogs arose from the wolf lineage in association with modern humans. The archaeological record puts the first undisputed dog at 14,700 years ago, found buried beside human remains, which is an interesting story in and of itself, because the evidence showed that the dog's neck was actually broken, which shows that by at least 14,000 years ago, humans and dogs were so closely associated that when the owner died, the rest of the people that were burying the owner wanted to bury the dog with the owner, knowing that it was a really important animal to that person in their life. So they actually made sure that the dog was dead and put it in there with the owner. Now, there are disputed remains that go back 36,000 years, and you can imagine why this would be tricky, because in the early days, a domesticated dog probably didn't look superficially that different from a wild wolf in terms of the types of remains we're going to find after 36,000 years. So the bone structure is going to be similar enough that it's going to be hard to really say if it was a dog or still a wolf. But either way, both of these dates are old enough that we're looking at a time when humans were still living in small tribes of hunter-gatherers and not farming or using agriculture. And this also suggests that the dog was probably the first domesticated species, because many other domesticated species came about with agriculture. So things like cats and horses and cows were necessary for agriculture to be successful, but dogs not so much. The general kind of thinking is that you would have groups of wolves that would be living in close proximity to groups of human hunters, and human hunters would bring down an animal, and the wolves would want to scavenge from that animal, and the wolves that were nice, so friendly, and didn't try to bite or snap or snarl, would come close enough to the fire that all these humans were huddled around and get a little scrap of meat. And if you kind of separate out the groups of really aggressive wolves that didn't want anything to do with humanity, and then only breed within the groups of wolves that were willing to come close to humans, you can kind of imagine how you would eventually get dogs. One of the coolest things about modern dogs is that because they've been selectively bred for so many generations, there's a huge variation in appearance, size, and weight. So much so that the genus Canis is actually the biggest range of sizes within a single genus of any organism on Earth that we know of. So the smallest dogs are only a couple inches tall and a couple inches long and only weigh a couple ounces, whereas the largest dogs can be in the hundreds of pounds range and stand 3.5-ish feet off the ground at the shoulder. Thanks to their close association with humans, dogs can now be considered the most widely abundant carnivore in the world. And I think it's a good thing, because dogs are great. Who doesn't love dogs? They're the best. Doggos forever. So that's a little bit about dogs and wolves and where they came from and where they are today. And so celebrate the puppers in your life. And we will see you very soon with another segment. So stay curious, because there's a lot to learn.
Organized curiosity. So I was sitting here trying to think about whether you were talking about cells or what. I don't know. I need to listen to it again to actually figure out, because I was so hung up on the fact that you were able to get an academic abstract out of the most common words, which leads me to believe that we as academics are unnecessarily verbose and use large words in order to impress. And guess what? What you read sounds equally as complex, but you used words that average people can understand. And I actually love it. Now, that may not fly the next time I try to write an abstract for a paper I'm going to submit, but I really love that. And I think that they're doing an amazing thing by having people use the most common words. Cheers.
Hey out there, everybody. It's Ryan from Organized Curiosity, and Tachi called in with a response to my somewhat silly segment about the Up-Goer 5 Abstract Challenge. And she was a really big fan, so I appreciated that, because I tend to get a little self-conscious in wondering whether or not this exercise is legitimately useful. And one of her points was that maybe scientists really do need to think about how verbose we are and try to simplify our language as much as possible. One of the best books on writing I've ever read that wasn't even necessarily a science writing book was The Elements of Style by Strunk and White. It's a classic text, and it's really short, and it's, fortunately, otherwise I think a lot of their lessons would fall flat, very, very readable. Like it actually is a page turner, even though it's about the concept of writing. One of my favorite rules from that book is to use as few words as possible to get your point across, and to use the shortest words possible to get your point across. And one of the ways that they both make that point and then also undermine themselves is with the 2-word rule, eschew obfuscation, which means avoid being confusing. The problem is those 2 words are kind of confusing and kind of hard to say one after the other. So there are cases where you have to use more words to get your point across than less words. Now, one of the things I like about the way that scientists talk is that we have a lot of 50-cent words that seem highfalutin, but I would argue that they're highfalutin only because they're not commonly used in everyday English. yet they do provide a very precise or information-dense way of getting the point across. The problem is when we talk that way to people not in our field and fail to define the terms as we use them and just assume that the people can keep up, because that's not a reasonable thing for anyone in any profession to do when that profession utilizes specialized language which has very specific definitions within that field. It's annoying when your car mechanic does it talking about parts of your car you don't know what they mean and where they are, and it's annoying when scientists do it when talking about their research. So I think it's a thing that we have to all be conscious of when we're talking to people who are outside of our sphere of the work we do. One thing I forgot to mention is that there is a text editor online. So Tachi, if you're actually interested in writing your next abstract using only the 1,000 most common words, they've made it very easy to do that. If you go to Splasho, that's splasho.com/upgoer5/, it's just a little text box, and it will underline any word that you shouldn't be allowed to use with the 1,000 most common words in red, so you can see it and work to get your story figured out using just those most common words. So if it's an experiment that other scientists out there who listen to this show want to try, or anybody who does anything specialized on a day-to-day basis, go give this a shot. Type a quick paragraph about the work you do, and then call into the station and read it out to us. So thanks again for giving me another chance to talk about this, Tachi, and feel free to call in with any other comments that you have. All right, thanks for listening. Be sure to favorite the station to get updates every day as I put them out, and remember to stay curious out there because there's a lot to learn.
All right, so I was kind of rushing through those first two segments, but things a little slowed down now. I was trying to get them all done while Julie was out for a walk, while I was working on dinner, but Julie and Clementine came back. Dinner needed to be finished, so I had to run off and take care of that, and now it's a few days later. Turns out you have to take dogs outside again. Eventually they have to pee another time, and so they're out again, and so I have a few minutes to myself to get this last little bit recorded. Time enough for a quick PaleoPOW to end the show, end the year. I just want to thank everyone who supported the show throughout the course of 2020. It's been tough. I know we've talked on and off about our sort of philosophical reasons for not diving into too much COVID stuff. Hopefully once a significant portion of the population is vaccinated, and I don't know that we could return to anything that could be constituted as normal, but maybe at least we can talk about some of the really cool science that actually has happened in regards to the pandemic and the new vaccine technology that's been developed. There is some really cool stuff, and I think it's worth discussing. But let's get through the end of the year before we do that and come in fresh for 2021. I'm happy to report that we've already recorded the next episode coming after this. It's going to be a really fun one that Tim helped me out a lot with, and so I think you should all look forward to that. For my PaleoPOW this week, I would like to thank Zachary G for being a recurring donor. Again, this is not the new Patreon system. This is the old school PayPal system where people just gave us a little bit of money every week without even a promise of a reward, just a promise of another episode coming down the pike eventually. So Zachary G, thank you so much for contributing to the show in that way. We really appreciate it, and it's kept us going for as long as we've been at this. So that's just really wonderful and means a lot, especially in this holiday season. One of the things you'll notice if you go to the website, you'll see that instead of an Amazon link, we now have a bookshop.org link. And bookshop.org is a service that works with local bookstores to sell books online and then make sure that the local bookstores fulfill those orders and ship those orders out and send them to wherever they need to go. So obviously you can get a sense for who they might be competing with in this space, but we have signed up to be an affiliate with them. So if you go to bookshop.org/shop/science sort of, you can see our Science Sort of store page. I'm working on putting together a list of books by folks who have been on the show, Hopefully that will be a little bit more fleshed out, but it's the sort of thing where if you click through that link on our website or go to our shop page and then shop at bookshop.org, we will get a little bit of the money that you spend. So will a local bookstore, and so will the people who made and wrote and produced the book. So I think it's a good system. I'm really excited to support it. I hope you all will join in on making this a good system for us to get some books out there, get some science out there, but also help out the local bookshops that I know have been especially struggling this year, along with a lot of other in-person businesses. So give that a shot if you so choose, and we would really appreciate it. And give us some feedback if you have had experiences with Bookshop or want to tell us about some other way that we can be helping out local businesses during this time. Please don't hesitate to reach out. You can email us, PaleoPOWs@sciencesortof.com, or reach us at the contact page on our website, or call and leave us voicemail. It's been a little while. 321-PaleoPOWs at 321-725-3672. Oh, and if you haven't had quite enough of me talking to you about things that are sort of science and sort of silly, I do want to let everyone know that I was really excited to be a guest on the MonsterTalk podcast, a podcast I've enjoyed for many years now. And I was talking about the Loveland Frog, which was a topic I covered for one of the Skeptoid episodes I did. So they reached out to me, asked if I would want to go a little bit more in depth, talk about some updates to the story that have happened since my Skeptoid episode, and I did, and it was super fun. Had a great time talking with Blake and Karen. So go give that a listen. Links will be available in the show notes for this episode, or just search Monster Talk Podcast, whatever you get your podcasts, and look for the most recent one. Should be me. If not, try a different episode. There's lots of good ones there. Check it out. Enjoy it. And with that, thank you for listening. Hope you enjoyed this little anthology-style episode to round out the year. So I hope everyone has a good couple of days of the rest of the year. Looking forward to a brighter and more hopeful 2021. We will see you all, hear you all, listen to you all in the new year, and we will get through this next year together, hopefully with a few fewer curveballs thrown at us. All right. This is Dr. Ryan Haupt. I did it. I finished the PhD. Got to go get to work on other stuff. Signing off for 2020, signing off for science, and signing off for Sort of.
Visit sciencesortof.com for show notes, links to all the stories we talked about, and ways to interact with the hosts, guests, and other listeners. Science Sort of is brought to you by the Brachiolobe Media Network of podcasts, with audio engineering by Tim Dobbs of the Encyclopedia Brunch podcast. That's all for this week. See you next time on Science Sort of.
That was just a weird little riff on the end that I did, but I'm alone, and it's fine. All right, bye.